So this lesson is lesson 5, multiple alleles. And we will look at how multiple alleles are being expressed in a gene. So a gene not only has two, it has more than two alleles. And so I think the definition for multiple alleles is pretty clear. It refers to a situation where there are more than two alleles. So maybe there are three, or maybe there are four, or maybe even five alleles for a gene, not just two. While a gene with only two alleles can display only one of the three dominance patterns, like, for example, the complete dominance, incomplete, and codominance. But since a multiple allele situation is more than two, there are options available. You can either stick to one of these dominance patterns. For example, all of them is complete dominance. One of them is completely dominant over all the other ones. Or, or you can have a mixture where one of the pair or one of the combination can be a complete dominant scenario, and then some of the other two alleles could be a codominant, or some of the other two could be incomplete dominance to each other. So there are options available. You can mix it up. One of the very classic example, and we actually have already looked at one, would be the human blood type, where you can have three different there are three different alleles. You can have an A or B or an allele for O. Now, what I want you to do is have a think about how can you tell if a scenario or a situation is multiple allele controlled, or is it just two alleles? And the easiest way would be to think about the number of alleles and the number of phenotypes available for a gene. So based on the number of possible phenotypes, you can actually kind of tell if this gene is just a simple two allele scenario, or is it a multiple allele scenario. So right here is a table in your notes, and I want you to give me the number of phenotypes for each scenario. So the first scenario would be if you only have two alleles in the gene and the dominance pattern is complete dominance. How many possible phenotypes are there? Let's say you have allele A and allele B, and allele A is completely dominant over allele B. And then figure out the rest of them, all right? So pause the video to do that. All right. So if you have done things completely, you'd have some numbers like these. So complete dominance is when one of the alleles completely dominant over the other one. And since you only have two alleles, that means the number of phenotype, it's going to be two. You can either get a dominant situation, or you can get the phenotype for the recessive if you get two of the recessive alleles. Whereas incomplete dominance, there are three phenotypes. You can either get a phenotype that displays the dominance, one of the allele, and then you can have a scenario that displays the other one, or you can have a phenotype that displays the mixture or the mixed up effect or the combination effect of the two alleles. So the number of phenotypes would be 3. And then for codominance is similar. You can have one displaying the allele, and you can have two displaying the other allele, and then you can have a third phenotype that displays the effect when you get two of the different ones. So you can have 3 phenotypes as well. However, if we move beyond just having two alleles, And the dominance pattern is a mixture, just like the blood type, where these two are completely dominant over O, and these two are co-dominating with each other and generates a blood AB group for you. Then what you'll get is you have 4 phenotypes. So I think it's pretty clear that once you move beyond 3 phenotypes, so having 4 or more phenotypes available and observable in a situation, then it's certain that the number of alleles in a gene is not just 2, but 3 or more, right? Okay, let's look at an example together. Coat color in rabbits. Now, coat color in rabbits, there are actually 5 different coat colors in rabbits. You can have one of the wild types, or we can call it brown. And then you can have a coat color that's called chinchilla. Chinchilla means that you can have fur, but the base of your fur, the part that is close to the hair follicle, is actually white. And then the tip of the fur is a black or dark gray color. And that's going to give you a chinchilla. And then you can also have a Himalayan, which means actually your body is kind of white. And then only the tip of your nose and your ears and your limbs are of a dark gray color, like the gray here. And then you can have a light gray color where the entire body is light gray color. So the entire fur from tip to the part where it's close to hair follicle is entirely just light gray. And then you can have albino, where the entire rabbit is white and the eyes are red. And these are the possible genotype to produce each of these phenotype. And what I want you to do is fill in this table right here at the bottom. I want you to observe the relationship between the possible genotype and the phenotype. So they're right here. And I want you to be able to sort out the dominance pattern that you can see between each of the combination between the alleles, right? Because this is the key step to doing a multiple allele question. Whenever you see a question with 4 or more phenotypes, you know it's a multiple allele situation. And the next thing you always do, is look at the relationship between each of the alleles and be able to determine it first before you can do anything really. So pause the video and complete this table. Okay, assuming that you've done it and tried it on your own, here's the time for answers. Now, if you look at the table, you can see that whenever a C or a big C is there, for example, this situation, you'll definitely get a brown rabbit. And that shows you that the big C is dominant over any other alleles. If it's with a chinchilla allele, then it's still a big C. For example, this situation here, the little CH represent chinchilla variant or the chinchilla allele. And then the C with the H represents the color or the coat with Himalayan phenotype. And so you can see whenever C is coupled with any of the other ones, it is shown through. So this is the dominant absolute boss in the gene. And then between the C and the H, because we have 2 other variations, we have the CH and then we have the H, right? So whenever they are together, and this is the only phenotype that is being generated, if you ever think about it, what type of relationship do you think this one has Would it be complete, incomplete, or codominant? Now, my answer would be incomplete dominance, because if you have a complete dominance, not an incomplete, what you'll see is patches of white and patches of this darker gray chinchilla color. But instead, the entire rabbit is actually a light gray color. So it shows you that some of this effect and some of these effects are being mixed together and combined, right? Instead of both of them completely showing themselves. So it's kind of this scenario right here. You get a mixture of color, not patches of color, all right? So if you get a mixture of color, it's incomplete dominance. And then last, we have to sort out the relationship of the small little c and all of the other colors. And what you see is that whenever a small little c for the albino is present, right here and right here, the albino color doesn't come through. So it shows you that the small little c is actually recessive to any other color. So the brown can dominate over her, the chinchilla can dominate over her, the Himalayan allele can also dominate over the albino color. And there we go. After you've sorted this out, then the rest of the Punnett square would be done just like any of the other Punnett square you've done before. You write down the genotype for the mom and genotype for the dad, put them into the Punnett square, and then you'll work out the probability of the child based on the different combinations you get in the Punnett square. So what I want you to do is complete the rest of your notes, the practice questions, and then the answers will be uploaded later as a video.